Hello, everyone. Welcome to today's live broadcast, Natural Human Gene Knockouts and the Discovery of New Drug Targets. I am Judy O'Rourke of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. We have a few important announcements before we begin. This is an educational webinar and thus offers free continuing education credits. After the webinar is over, and to get your credits, click on the CE button located in the bottom left-hand corner of your web page. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you have any technical problems viewing or hearing this presentation, please click on the support button at the top right of your presentation window or submit your problem through the green Q&A button lower left. I would now like to introduce today's speaker, Rick Dewey, MD, Head of Translational Genetics at the Regeneron Genetics Center. Dr. Dewey received his AB in Chemistry and Physics from Harvard University and MD with concentration in Cardiovascular and Pulmonary Sciences from Stanford University. He received clinical training in internal medicine and cardiovascular medicine at Stanford Hospital and Clinics and research training in human genetics via the Stanford Clinical Investigator Pathway. His research work has focused on gene discovery in familial and complex cardiovascular disease using high-throughput sequencing and application of exome and genome sequencing in clinical care. He joined the Regeneron Genetics Center, a wholly owned subsidiary of Regeneron Pharmaceuticals, in 2014 and currently leads the Translational Genetics Group. I will now turn it over to Dr. Dewey for his presentation.
Hi, good morning, and thank you, Judy, for the kind introduction, and to the organizers for giving me the opportunity to talk about some of our work that we've been doing at the Regeneron Genetics Center, focusing specifically on natural human gene knockouts and the application of exome and genome sequencing for drug discovery purposes. Thank you to all of you for tuning in, particularly those folks on the West Coast for whom this is happening at quite an early hour. So today I'll talk a little bit about some of the stark realities of therapeutic development in 2015, 2016, and the ways in which whole genome sequencing, whole exome sequencing can be leveraged to improve the discovery and validation process for new therapeutic targets. We'll touch on some of the work that we're doing at the Regeneron Genetics Center and focus on a few examples, specifically within the lipids and cardiometabolic space, of the way in which that we are leveraging whole exome sequencing, in particular, in large populations spanning family-based designs for human genetic purposes, all the way through to large complex or interrogation of complex disease and population case control studies, as well as large clinical cohorts that we think are emblematic of the promise of the Precision Medicine Initiative. So the reality, of course, in 2015 is that increased investment in research and development in the pharma industry has not led to a dramatic increase in the number of new molecular entities. The challenge here, of course, in bringing these molecules to market is that over 90% of molecules that enter phase 1 clinical trials fail to demonstrate sufficient safety and efficacy to eventually gain regulatory approval and make it to the patients that need them most. Most of these failures are now occurring in phase 2 clinical trials, 50% due to lack of efficacy and a small proportion due to toxicity. And the challenge that we face is that preclinical models that we've relied on for decades and have espoused here at Regeneron and in other places may be poor predictors of clinical benefit. It's now been demonstrated that compounds that are supported by human genetics evidence are substantially more likely to succeed. And so one of the things that I'll talk about today is the way that human genetic evidence can be incorporated into the drug target discovery and validation process. So there are numerous examples of ways in which human genetics have pointed the way, either prior to development of therapeutics or subsequent to the development of therapeutics to targets that are efficacious in humans. One of the canonical examples we've pointed to in recent years has been that of PCSK9, and I'll see if I can use my pointer here to point that out, which is a poster child for the way in which human genetics has pointed the way to a therapeutic target. Based initially on the observation, I'll talk about this in some subsequent slides, that gain-of-function mutations segregated in individuals harboring autosomal dominant presentations of familial hypercholesterolemia, a very severe early onset form of high cholesterol that's associated with substantially increased morbidity and related mortality due to atherosclerotic cardiovascular disease. Subsequent to that, observations made by Jonathan Cohen, Helen Hobbs, and others solidified the other end of the story, or the other end of the spectrum, in which loss of function mutations, beneficial loss of function mutations in large human populations were found to confer favorable lipid profiles and protection from cardiovascular disease. And this has really spurred the development and a race to the finish line now of 3 molecules that target PCSK9, which internalizes the LDL receptor, thereby reducing hepatic LDL uptake. There are a few additional examples of this now in recent years, including that of ApoC3, really starting with observations in the Old Order Amish, a founder-like population that by virtue of geographic and reproductive isolation harbors alleles at a surprising frequency, including the apolipoprotein B mutation R3527Q that is associated with an autosomal dominant form of familial hypercholesterolemia. In that population, observations made by Alan Schuldiner and others at the University of Maryland found a stop mutation, a premature stop mutation that abrogates apolipoprotein C3 function, thereby reducing triglyceride levels in humans. Subsequent to that, a number of investigators, including Stig Katharesen, investigators at Copenhagen, Ann Tadger Hansen, and Baron Nordeskard, were able to demonstrate that these same mutations in very large populations conferred not only favorable reduction in triglyceride levels, but also approximately 40% reduced odds of coronary disease. And this has spurred, again, antisense molecules to apolipoprotein C3, really opening the door in a big way for triglyceride-lowering therapies that might have a beneficial effect in humans with respect to atherosclerotic cardiovascular disease. Here, just summarizing again these 2 stories in which the initial observation often was made between a half and a full decade prior to a clinical proof of concept, by which gain-of-function mutations or loss-of-function mutations that were associated with either unfavorable or favorable clinical manifestations in human populations ultimately led to the development of these 2 therapeutics. The goal of what we do is to really accelerate this process by bringing the human genetic discovery process in-house, nested within an early phase group among research and development activities here at Regeneron, and accelerate the process from initial genetic insight to therapeutic development and deployment within the clinic. So we have really 3 main goals here for human genetics, that of indication discovery, that of new target discovery by identifying new drug targets and pathways via human genetic observations. And I'll tell you a few stories about that related to identification of loss of function mutations that confer a favorable clinical profiles in human patients, as well as the identification of new biomarkers to predict a response to disease, either adverse or beneficial, and help to further stratify patient populations to those that are most likely to respond favorably to our drugs. And these are the 3 tenets for the way that we are using human genetics here at Regeneron to guide our drug discovery and development process. Our leadership team here consists of Alan Schuldiner, who, again, as I mentioned earlier, came to us from the University of Maryland, Eris Barris, who has been a longer time Regeneron employee, and previous to this was at Duke, Ingrid Brecki, who just joined our team and leads our analytical genetics group. I lead our translational genetics group here at Regeneron, and previously was at Stanford. Eris Economides, who is heading our functional modeling group, Omri Gottesman, who leads our clinical informatics group and came to us from Mount Sinai, and John Overton and Jeff Reed, who lead our sequencing and lab operations and genome informatics groups, respectively, coming from Yale as well as from Baylor. So the way that we've been approaching the identification of and validation of new drug targets using human genetics is to employ a multi-pronged approach in which we fully espouse an approach in which integrated human genetic studies across genetic trait architectures, from the smallest family-based studies of individuals segregating and dealing diseases all the way through to general population studies in which we are gathering phenotypes more or less by convenience from large clinical populations will help us to fuel genomic discovery. I'll tell you a little bit about the latter and what we think of as a groundbreaking collaboration with the Geisinger Health System located in central and northeastern Pennsylvania. Many of you, I'm sure, have heard about the Precision Medicine Initiative announced by President Obama in the early part of last year, which has proposed a very bold goal of sequencing approximately 1 million volunteers for a research program which is very broad-ranging. And this involves linking genetic data to electronic health records as one component of a broad program that aims to personalize the approach to medicine and really improve the precision by which we deliver care. The primary tenet here is that understanding how genomic variation and other health factors influence human health and disease can allow us to better treat patients in an individualized fashion. And the goal here is to build a very comprehensive knowledge base for implementation of precision medicine that we hope will not only improve our understanding of the fundamental basis of human disease in large populations, but also perhaps spur the development of new therapeutics that are targeted to specific populations. We have with Geisinger embarked on a very similar program beginning really in the late phase of 2013 and drawing on their MyCode community health initiative, which is a very ambitious biobanking effort that has to date enrolled over 100,000 individuals with a goal of applying genomic-based interrogation to identify new levers that drive disease, as well as perform genomic medicine implementation at a scale that has yet to be matched anywhere else. And so we have a goal at this point to sequence over 250,000 people within the Geisinger Health System and build what we believe to be one of the world's most comprehensive genotype-phenotype association resources using de-identified clinical data from all 250,000 people to aid our drug development and genomic medicine efforts. Geisinger will tell you a little bit about in some subsequent slides, but it is a very large integrated health system that exists in central and northeastern Pennsylvania and is advantaged for this type of research by several things, including cradle-to-grave records for over 2.5 million patients and approximately 1.4 million of whom are very active participants within the health plan. This is a very large population-based study that aims to sequence over 250,000 participants and integrate genetics with electronic health record-based clinical phenotype information. The Geisinger Health System, as I mentioned, has a catchment area which exists in central and northeastern Pennsylvania and aims to connect these cradle-to-grave electronic health records with genomic data, primarily exome sequencing and array-based genotyping. Approximately 100,000 of these patients have been consented to date, and approximately 60,000 have been sequenced and genotyped at the Regeneron Genetics Center. In addition to a large unselected patient population, there are several targeted recruitment efforts, including efforts to engage participants in a cardiogenomics protocol, enrolling patients from the cardiac catheterization lab who have angiographic data about their coronary anatomy, as well as physiological data about cardiovascular function, as well as a large ascertainment via the bariatric surgery clinic, which is the largest, one of the largest research studies of its kind in the United States. The Geisinger Health System, as I mentioned, is advantaged by being one of the earliest adopters of electronic health records in the United States, adopting the EPIC platform in 1996, and having a relatively stable patient population in which the participants in their health plan receive all of their primary and subspecialty care via the health system. And so this is a relatively complete accounting of their medical care that is captured in electronic health records that we identify, that we receive de-identified forms of. Why is sequencing at scale required for this type of effort? For what we are focusing on, which is functional coding variation within predominantly exonic regions of the genome, turns out that this variation, not a surprise to anyone who's been involved in this science for some time, is exceedingly rare. And so this is some data describing sequence variants identified using whole exome sequencing of the first 50,000 participants in the Discover program that we've embarked on together with Geisinger, in which you'll appreciate via the site frequency spectrum here on the right that more functional variation, in particular loss of function variation, is enriched for exceedingly rare alleles. And approximately 50% of the variation that we are observing of this class occurs only once within the data set. We've discovered approximately 4 million single nucleotide variants and approximately 220,000 insertion deletion events in the protein coding regions of each of these 50,000 individuals' genomes, and approximately 175,000 predicted loss of function variants, thereby producing a vast reservoir of functional variation for us to interrogate with respect to its effects on clinical phenotypes within this group of individuals. Again, not surprising data. This has been observed by others, including Dan MacArthur and folks who have been involved in the Exome Aggregation Consortium study. But each Discover participant here harbors approximately 20 predicted loss of function variants, the majority of which occur in heterozygous state, and approximately one rare homozygous loss of function variant has been observed per individual, thereby providing, again, a vast reservoir for predominantly heterozygous observations in terms of their association with clinical phenotypes. What does the coverage of gene regions look like with loss of function variants as you sequence deeper into this population? Here we have the accrual of loss of function variation as a function of population size on a per gene basis. And what you'll observe is that even at this sample size, we have near complete coverage of autosomal protein coding genes with loss of function variants in one or more individuals in heterozygous state. And approximately 47% of genes at this sample size harbor heterozygous mutations in 20 or more individuals, providing a resource at least for quantitative trait association testing, and increasingly now for binary trait association testing to identify disease associations that might be a good mimic of what antibody blockade or other therapeutic antagonism might do in a human. Connecting this, of course, to the clinical phenotype data is really what is necessary for phenotypic association discovery. So here's just a very high-level view of the Geisinger Clinical Data Warehouse, which is a live phenotype database in which we receive clinical phenotype information on a rolling basis. This is a very large repository of information with approximately 40 tables, 1.5 billion rows, that grows by 308 million rows. That includes 308 million rows of lab results and grows by 520,000 rows per week. This database is represented in de-identified fashion as the back end to Epic, and we receive this clinical information and in concert with our Geisinger colleagues transform it into binary and quantitative traits that allow us to do association testing with our genetic data. Just again, another top-line summary of the clinical data housed within the Geisinger Health System. There are a very large number of high-quality EHR-derived phenotypes that are available for discovery purposes. You'll note that on the top left here that the median number or the median amount of data within the health care system here is approximately 8 years of data for each participant that's been sequenced. Common things being common within this healthcare system, we have a very abundant representation of common complex diseases, including hypertension, hyperlipidemia, coronary artery disease, acute myocardial infarction, and the like, and relatively abundant representation of laboratory values with repeat measures that allow us to understand over the course of a lifetime the genetic influences that determine laboratory and other traits within the healthcare system. As a supplement to what's been gathered in the course of routine preventative care, we also have trait values that cover other ancillary data sources, including echocardiography, electrocardiography, ocular measures, PFTs, DEXA data, and the like. And all of this really makes us a very rich repository of phenotypic information for association discovery that we hope can drive our efforts. So as proof of principle, I'm showing you here associations between loss of function variants and genes that encode lipid-lowering drug targets, and here hoping to convince you that they recapitulate the established pharmacologic effects of these drugs, including things that are currently in early phases of clinical development, including ATP citrate lyase inhibitors, a story I'll tell you a little bit about here in subsequent slides around an inhibitor of angiopoietin-like 3, which is an endogenous inhibitor of lipoprotein lipase. And what we've observed here is that nearly all of our targets harbor loss of function variants, which at least nominally, according to nominally significant thresholds, recapitulate the drug effects of therapeutic antagonism of these targets in human populations. We've been able to subsequently evaluate some of the targets that are currently in our early phase discovery or development pipelines. And here I'm going to show you some data around ANGPTL4, which is an endogenous inhibitor of lipoprotein lipase. Lipoprotein lipase is one of the canonical enzymes involved in triglyceride metabolism and acts in various tissues to partition free fatty acids to oxidative and storage tissues according to metabolic need. In concert with our Geisinger colleagues, we evaluated the relationship between a low-frequency missense mutation in ANGPTL4, the E40K mutation, which has been previously demonstrated to abrogate the ability of ANGPTL4 to inhibit lipoprotein lipase, as well as other putative loss-of-function mutations that were found within our cohort and associated with lower levels of triglycerides and higher levels of HDL-C. What's been clear from previous work is that these mutations are robustly associated with triglycerides and HDL levels, but what has been less clear is whether or not that lipid profile will translate into a reduction in atherosclerotic cardiovascular disease risk. And so we asked that specific question both of the E40K mutation as well as of the heterozygous inactivating mutations that we observed within our data set, and found that the E40K mutation among approximately 10,000 coronary artery disease cases defined angiographically and approximately 30,000 matched controls was associated with about a 20% reduction in the odds of coronary artery disease, and heterozygous inactivating mutations were associated with a more dramatic reduction in odds of coronary artery disease, thereby providing additional allelic evidence that antagonism or genetic antagonism of ANGPTL4 might be beneficial with respect to atherosclerotic cardiovascular disease, and also, I think, providing some additional evidence that modulation of triglycerides might have a beneficial and potentially causal effect in reduction of cardiovascular disease risk in humans. We subsequently extended that observation to antibody treatment in non-human primates, as well as in mice who are homozygous for a humanized form of the ANGPTL4 gene. And that's the panel here, A, here on the upper left, in which antibody treatment of these mice was associated with pretty substantial reduction in triglyceride levels. And then among cynomolgus monkeys on high-fat diet, as well as rhesus monkeys with hyperlipidemia, antibody treatment was similarly associated with substantial reductions in triglycerides. Interestingly, antibody-based inhibition of ANGPTL4 appears to reduce triglycerides to near normal levels despite or irrespective of the starting point for triglycerides. So in a single monkey with severe hypertriglyceridemia, here in the lower right-hand panel, we observed a reduction in triglycerides from approximately 2,000 milligrams per DL to near normal levels, suggesting that this might be highly efficacious for even the most severe forms of hypertriglyceridemia. We've then extended this observation to one in which we asked the question about E40K and risk of type 2 diabetes. Here showing data from our population, the Geisinger Health System population, and approximately 11,000 type 2 diabetes cases defined via an electronic health record-based algorithm that incorporates diagnosis codes, medication use, and laboratory values, as well as now in approximately 62,000 additional cases and 290,000 additional controls, a robust association between the E40K mutation and reduced risk of type 2 diabetes, perhaps providing a link between triglyceride metabolism and insulin sensitivity or insulin release that demonstrates a benefit both for triglyceride lowering and reduction of atherosclerotic cardiovascular disease risk, as well as reduction of type 2 diabetes and improved glycemic control. We've been able to use our in-house mouse resources to, again, confirm this observation. Here showing on the left-hand panel that fasting glucose levels in 67 black 6 mice who were deficient for the ANGPTL4 gene were improved in comparison to wild-type mice. And subsequently that oral glucose challenge, as well as insulin tolerance testing, demonstrated improved improved insulin sensitivity and improved postprandial glucose tolerance in these mice, perhaps demonstrating that ANGPTL4 is mechanistically linked to improve glucose control. This is intriguing because this is one of the first genes that we are aware of that, when inactivated in humans, leads to a combined effect on reduced odds of type 2 diabetes and reduced risk of atherosclerotic cardiovascular disease, and we think might represent a very intriguing target with respect to metabolic disease risk reduction in humans. One of the concerns around therapeutic manipulation of ANGPTL4 has been an observation that ANGPTL4 deficient mice, or antibody treated mice and non-human primates fed a high-fat diet had developed mesenteric lymphadenopathy, which in the case of ANGPTL4 deficient mice had been severe enough to lead to chyloscystitis and in some cases fatal peritonitis. One of the intriguing questions is whether this preclinical observation will translate to humans. And this is of critical concern to us as we consider whether or not to push forward the ANGPTL4 molecule as a therapeutic target. And it turns out that we have a relatively reasonable number of homozygotes for the K40 mutation, which abrogates the ability to inhibit lipoprotein lipase, that are walking around free living within the Geisinger Health System, and the ability to interrogate what they look like from the standpoint of clinical concerns that mimic the abdominal lymphadenopathy and other abdominal pathology seen in preclinical models. What we've been able to observe thus far, at least from electronic health record-based codes, is that they do not appear to manifest ICD-9 codes that correspond to disorders of the lymphatic system, ascites, peritonitis, or even more general abdominal complaints at a higher rate than non-carriers within our population. And importantly, in deeper chart review of these individuals, 5 incidentally had CT abdominal imaging, and 4 or 5 on that imaging study had explicit mention of normal abdominal lymphatics, and 1 of 5 had no mention of abdominal lymphatic abnormalities. This thereby, I think, is an intriguing set of observations that at once potentially validates this as a drug target for metabolic disease risk reduction in humans, and raises the question of whether this is therapeutically approachable, even in light of the preclinical findings in non-human primates and mice. One of the advantages of the Geisinger Health System collaboration is the ability to perform genotype-based callback studies, given that these individuals are all consented for that activity, and that's something we're currently designing. We have a related molecule that's currently entering the clinic that targets angiopoietin-like 3, which has been found in kindred segregating familial combined hypolipidemia, absent the hepatic triglyceride accumulation that is typically associated with familial A-beta or hypolipoproteinemia. What has been clear from previous work is that ANGPTL3 loss of function mutations, both in large populations as well as in families, some work done by Nate Stitzel and colleagues, as well as by Jonathan Cohen and Helen Hobbs again, have been associated with a combined hypolipidemia profile that canonically involves depression of all lipid fractions, HDL, LDL, as well as total cholesterol, as well as triglyceride-rich lipoprotein levels. What has not been clear, again, is whether or not that translates into clinical benefit, and this is of utmost importance as we move our ANGPTL3 inhibitor into the clinic. We are now, I think, reasonably confident that ANGPTL3 loss of function mutations, at least in the Geisinger population, are associated with lower odds of atherosclerotic cardiovascular disease as defined geographically, here demonstrating an odds ratio of approximately 0.6, demonstrating about a 40% reduction in odds of coronary disease, similar to that in our cohort observed for PCSK9 loss of function mutations. These variants, we've identified now approximately 12 of them, are associated with an approximate halving of circulating ANGPTL3 levels, as is consistent with full loss of one allele, as well as an approximate 28% reduction in triglycerides, 78% reductions in HDL and LDL, respectively, which are highly statistically significant. And this has translated, despite the HDL effect, into a reduction in odds of coronary artery disease in our cohort. If confirmed externally, I think this presents an intriguing opportunity for us as we consider positioning the ANGPTL3 therapy not only for hypertriglyceridemia as well as for other forms of dyslipidemia. So I'll transition now just briefly to some of our efforts for target discovery from target validation and show you some selected examples of loss of function variant associations with disease. And in this particular case, providing some evidence that we can recover even things that are canonically thought of as being Mendelian expressions of disease within large clinical populations, namely loss of function or truncating variants within Titin, which are robustly associated with primary cardiomyopathy within our cohort, BRCA1 associated with hereditary breast and ovarian cancer, NOD2 associated with inflammatory bowel disease, NF1 associated with neurofibromatosis, and a host of other disease conditions that really lie at the intersection between what we could typically think of as monogenic or oligogenic disease and complex disease traits. We have a few other observations that we're currently tracking down that are novel, that are potentially quite impactful from the standpoint of developing new therapeutics, including one that appears to be associated with reduced odds of alcoholic liver damage. As I mentioned, the Discover collaboration with the Geisinger Health System is really dual purpose. We have a primary interest in using this data resource for drug target validation and drug target discovery. Geisinger has, I would say, an equally important interest in understanding what genomic medicine implementation might look like in the 21st century. And so really to address this particular question, what if everybody's genome is available in their medical record? And so they've embarked on a very bold initiative to engage individuals within the Discover program, enrolling in the MyCode Community Health Initiative, who are harboring mutations that appear to be clinically actionable on the basis of clinical presentation, pattern of inheritance, and the opportunity for early clinical intervention that might mitigate morbidity and mortality in this population. As I mentioned, the population is one that is stable and has lived here for several generations in many cases, and also has substantial familial substructure. And that leads to some interesting observations about alleles that segregate within families here in a way that might not be expected in a completely unrelated population. And so here is one example of that. For one of the 27 disease conditions for which the Geisinger Return of Results effort, the Genome First program, has been designed, namely familial hypercholesterolemia, we've identified a large exonic duplication in the LDL receptor that appears to be segregating in individuals displaying a familial hypercholesterolemia-like phenotype. in reconstructing these pedigrees from genetic information alone, we've identified a recent common ancestor approximately 6 to 7 generations ago within the Geisinger Health System that links all of these individuals. These individuals here have a very early onset cardiovascular disease and very high levels of total and LDL cholesterol that place them at substantially increased risk for the development of heart attacks and other cardiovascular disease conditions such as heart failure subsequent to heart attack. And there's a pretty substantial opportunity for early clinical intervention in the form of highly efficacious lipid-lowering therapy. So one of the primary programs here is to engage these individuals and understand what early clinical intervention and early identification of these patient populations might yield in terms of clinical benefit for this group. And so I'm showing you now some data on the prevalence of familial hypercholesterolemia as defined clinically, as well as on the basis of genotype alone via mutations in the LDL receptor, the apolipoprotein B gene, as well as PCSK9 within the Geisinger population, and which have identified, I think, a higher than had been previously appreciated prevalence genotypically of familial hypercholesterolemia in this population, with approximately 1 in 215 individuals here harboring a mutation that has been canonically associated with familial hypercholesterolemia. Not surprisingly, there's a higher prevalence of these mutations among individuals ascertained via the cardiac catheterization lab, with about 1 in 214 individuals ascertained via that setting harboring a mutation in one of these genes that has been canonically associated with familial hypercholesterolemia. As has been previously demonstrated by others, the presence of severe hypercholesterolemia is in itself not sufficient for the diagnosis of a familial form of hypercholesterolemia, or very severe hypercholesterolemia here. And this is demonstrated by the observation that among individuals with LDL-C levels above 190, which would qualify as severe hypercholesterolemia, only 1 in 40 of those individuals within our healthcare system harbors a mutation in one of the canonical familial hypercholesterolemia genes. Turns out that genotype based refinement of clinical recognition of this disease can certainly increase can certainly improve diagnosis, and so among individuals who are very positive, there's a higher proportion of individuals who meet clinical criteria or for familial hypercholesterolemia, and this is of course important because of the observation that that genetically identified patients with familial hypercholesterolemia have about 4.5-fold increased risk for premature cardiovascular disease, which is highly statistically significant. And then among individuals who present early with cardiovascular disease, there's an approximate 10-fold increase in risk associated with the presence of one of these mutations in this population that appears to be independent of the level of total LDL cholesterol, thereby suggesting that there's a substantial opportunity for early intervention here within this population on the basis of genetic information that could improve clinical care. Familial hypercholesterolemia is not often adequately treated within this population. Only 61% of genetic FH patients in our cohort were on statin therapy at their most recent encounter, and only 30% had achieved LDL cholesterol goals of less than 100 milligrams per DL, which is consistent with some emerging data around clinically identified patients enrolled in the Cascade Registry that has been put together by the Familial Hypercholesterolemia Foundation. As a supplement to what we're doing within the Geisinger Health System, we have a pretty substantial focus on familial sequencing of families that are harboring rare, apparently monogenic conditions, including pulmonary arterial hypertension, familial cardiomyopathy, early onset severe obesity, and type 2 diabetes. And we view this as a primary engine that is relatively high yield for novel disease gene discovery and can really sort of illuminate fundamental human disease biology. One sort of canonical example of that put together in collaboration with Wendy Chung at Columbia University and Claudia Gonzaga-Jaregui, who's been leading our Mendelian efforts here at the Regeneron Genetics Center, is the implication of TBX4 now in multiple individuals with apparent familial forms of pulmonary arterial hypertension, an early devastating disease in which there is high pressure in the pulmonary vasculature that can lead to heart failure and early mortality. TBX4 has been identified previously in the context of small patella syndrome, and there have been some early insights that suggest that this might be a gene that plays a role in the development of pulmonary arterial hypertension. And here, Claudia and Wendy have been able to provide some solid evidence via segregation in multiple families that rare deleterious variants in TBX4 are an important cause of early onset pulmonary arterial hypertension, perhaps the most important cause aside from mutations in BMP2. So here are just some additional data solidifying that point, in which approximately 11% of our cohort harbors mutations in TBX4, making it the second most impactful gene in early onset pulmonary arterial hypertension following BMPR2. I mentioned earlier the work that Alan Schuldiner and others have done in studying the Old Order Amish. We believe that this is a high-yield approach for novel gene discovery in which taking advantage of genetic homogeneity, principles of drift, as well as recent population bottlenecks, which by virtue of geographic and reproductive isolation from these populations allow certain alleles to drift to higher frequency, and by virtue of some degree of endogamy within these populations allow them to exist in homozygous state. We have an ambitious program to put together a consortium of these populations. We have termed DRIFT, Discovery Research Investigating Founder Population Traits, led by Alan Schuldiner here at the RGC, in which we have embarked on a two-tiered approach to study these populations, which individually are homogeneous, but we hope to amass a heterogeneous menagerie of these populations. The first tier involves defining the allelic spectrum within and across these populations to identify interesting functional variants that might exist at higher frequency in these populations than others, and interesting collections of homozygotes that can be studied further with respect to phenotypic expression of traits of interest for us. The second tier would involve genotype-phenotype association discovery, and especially for these mutations that are highly functional, that are enriched in a given population, and the engagement of these populations in a genotype-first manner via phenotypic callback studies. Our foundational partner here is the Old Order Amish, and we have a very rich collaboration with the Clinic for Special Children, initiated by Holmes Morton, now run by Kevin Strauss and Eric Puffenberger, which has both served clinically this population and the Mennonite population, and also has engaged them in research efforts in which they've been very willing participants to engage in genomic studies of their population to understand the fundamental basis of devastating diseases among their people. We also are embarking on a collaboration with the Old Order Amish via the Amish Research Clinic, run out of University of Maryland, which is specifically positioned to do in-depth phenotypic characterization of that population. Just a specific example of the way in which Old Order Amish demography has increased the number of some minor allele homozygotes. About 3.5% of each Amish genome is autozygous here. There's finite population size, and this results in some degree of endogamy/consanguinity. The example here in the lower right is one in which an allele has drifted from 1% frequency to 5% frequency in the Amish, and by virtue of endogamy, the minor allele homozygote frequency has increased approximately 40-fold. So we are recruiting for this, and so if there are those of you out there who have studied these populations or have access to these populations and are interested in engaging with us in large-scale genetic interrogation of these groups, we are more than willing to participate in research collaborations by which you and we both benefit from the study of these groups. So just to sum up, human genetics have been integrated across our discovery and development pipelines here at Regeneron, and we really fundamentally believe that this can change the way that drug development is done. And we do sort of drink the Kool-Aid with respect to the use of precision medicine and genomic discoveries in drug development, and believe that genetic experiments of nature can point the way to these new therapeutic targets and guide our drug development efforts. fundamentally espouse an approach in which combining these approaches across genetic trait architectures and phenotypes can help catalyze discovery, and integrate this, of course, across functional biology, genomic or functional genomics, and clinical pharmacology to drive our translation of discoveries into therapeutics. Our guiding principles, of course, here involve the study of large general populations, the integration of both Mendelian genetics and family-based studies with studies of founder populations and other disease-specific and clinical cohorts of deep phenotyping, coupling that with very deep phenotypic data, including longitudinal data, as well as quantitative traits, and then emphasizing large effect coding variation as one way in which we can provide some highly interpretable insights. Challenges, of course, in our early efforts have been the power to detect rare variant associations is, of course, hindered by very rare functional alleles, and thus, large data sets are required for both our early discovery efforts as well as replication efforts of our novel gene discoveries. And rare variant interpretation, including missense and non-coding mutations, remains a substantial challenge. Most of the variation that has come out of the GWAS era exists in non-coding regions, and I'm sure you'll hear more about ways in which we can functionally interpret that in the course of the program here today. But it does remain a challenge with respect to identifying functional units that might be approachable therapeutically. We've had to develop de novo methods to handle emerging computational challenges associated with very large both phenotypic and genotypic data sets, including new ways of representing the data graphically that allow us to rapidly traverse both phenotypic and genotypic information and perform association discovery. And then, of course, plugging this in to a pipeline that is impedance matched to our discovery efforts with respect to functionally interrogating and understanding these human genetic associations is of utmost importance as we drive these insights to translational opportunities and potentially therapeutics. Lots of people to acknowledge the development of this effort, including our wonderful team here at the Regeneron Genetics Center, which includes both analytical and translational scientists, as well as heavy informatics support from Jeff Reed's team, our leadership team here that I alluded to earlier, our collaborators who, without whom none of this work would be possible at Geisinger, Columbia, and now 19 other collaborating groups that have worked with us to really drive forward the human genetic work that we do here at the RGC, and especially to the thousands of patients, volunteers, collaborators, and investigators who have worked more or less anonymously with us along the way to drive this work forward. So thank you very much for your attention this morning and tuning in, and I'm happy to take questions at this point.
Thank you for that informative presentation. Before we get started on the question and answer session, I'd like to remind our audience how to submit questions. You can submit questions by typing them in the Q&A box, which can be found by clicking on the green Q&A button at the lower left of the presentation window. We'll try to answer as many questions as we can. And our first question is, do you have any efforts to perform haplotyping and examining the allele-specific expression in your 250,000 cohort?
A very interesting question. Certainly, haplotype-specific effects, parent of origin effects, and phasing is I think are increasingly important as we go beyond observations about heterozygotes within our data set. We do have ongoing efforts to develop technology specifically to molecularly phase and computationally phase our data, advantaged again by the fact that we have substantial familial substructure within our cohort that allows us to phase many of these alleles by transmission. With respect to allele-specific expression, there are efforts underway to perform sequencing-based expression profiling of certain tissues of interest to us, and coupled with that, allele-specific expression, I think, is one way in which we can gain insight into the expression of some of the more deleterious alleles that we find in our data set.
What about CHD genetic variants in other people and groups, or does Geisinger data have all groups?
Judy, would you please repeat the question?
What about CHD genetic variants in other people and groups, or does Geisinger data have all groups?
So the Geisinger data is predominantly from individuals of European ancestry, which we believe is one view of the slice of alleles that exist within humans that can be used for association discovery with respect to phenotypes. We have attempted to supplement that with other specific collections that have included other ancestral groups, including a large East Asian collection that is being sequenced in collaboration with Ida Chen and Jerry Rotter at the Los Angeles Biomedical Institute. These are individuals of predominantly Taiwanese ancestry.
What about hematological health conditions, as I didn't see the field in Discover?
So I believe the question was here again about CETP inhibition and the apparent failure of CETP clinically. And that is something that we are currently investigating. I would say that we don't have a clear answer yet with respect to the effect of CETP mutations and their relationship to coronary artery disease, in particular sort of a canonical way in which we hope that we can leverage human genetic data to answer a critical clinical question, which is, does the HDL lowering and HDL increase and LDL lowering effect are associated with reductions in atherosclerotic cardiovascular disease.
Does your data still correlate with the apparent failure of CETP? Sorry, where did that question go? Bear with me one second. Does your data still correlate with the apparent failure of CETP clinically?
Judy, thanks. I think I answered that question out of order here. That's something we're still investigating. At this point, the answer isn't clear.
While genetics may indicate whether an individual is predisposed to a condition, could you please address what other markers may indicate imminent onset of a disease, for example, lipidemia?
Very good question. So, of course, genetics isn't everything with respect to the development of disease, and in fact, in most cases, it explains only a very small proportion of either variants in a trait within a human population or variants in the risk for a disease. What we have attempted to do in our studies is to understand which genetic levers drive that small bit of variation, and from there make the leap to whether or not there are translational opportunities that might emanate from manipulation of those particular gene products. Within the Geisinger population, in addition to the sequenced cohort, we have access to clinical phenotype information from approximately 1.4 million participants in their health plan. And there are opportunities that fall from that to understand epidemiological and other relationships between environmental and other factors that help determine disease onset, and that's an area of active interest for Geisinger in particular.
Why would you use the TBX4 for gene implication? Why would you use the TBX4 for gene implication arterial hypertension?
I think if I understand the question correctly, there's a question about the relationship between TBX4 and pulmonary arterial hypertension within our family-based studies. In this particular case, we observed multiple individuals segregating TBX4 mutations who were also affected by pulmonary arterial hypertension. That segregation pattern was consistent with causality for this particular gene within that disease case.
Has the Geisinger system leveraged this data to apply pharmacogenomics?
Geisinger is absolutely developing platforms that will use genetic information to guide pharmacological therapy, canonically referred to as pharmacogenomics. One of the challenges there is whether is to identify the right way to incorporate pharmacogenetic information into routine clinical care in such a way that it is not disruptive to that clinical care and also has a substantial enough impact on disease endpoints to make the use of that information meaningful. There are guidelines fortuitously that have been developed for pre-existing clinical information, for pre-existing genetic information, and the way that can be used to guide pharmacological management of patients, the CPIC guidelines that I would refer you to, that have at least formalized the way in which this information can be incorporated. And Geisinger is currently in the process of considering the best way to deploy that clinically.
In the systematic ascertainment of founder population studies, did you find first or second tier harder to study and gather information from?
Very good question. The second tier studies involve gathering in-depth clinical phenotype data from these participants, and so that is, has been a more challenging mode of collaboration for us to engage in, and I think that that is something that we hope will improve as we sequence deeper into these populations, find more interesting collections of these alleles.
Does the Geisinger database have karyotype testing data?
Outside of the routine clinical applications of karyotyping, we do not routinely perform karyotype testing on the participants in the Discover population.
What happens to the database of volunteers who may have died during the data collection? The death could be attributed to the gene being observed, or the volunteer might have died naturally. I'm not sure if that's a comment or a question.
Interesting question. The volunteers who have passed during the study and have consented to the use of their genetic and clinical phenotype information continue to be used in the course of the study for phenotypic association discovery. One of the most challenging things to do is to identify genes that are associated with longevity, which could be a corollary to your question here. That has been a long run for many folks who have attempted to find single levers. As it turns out, it probably has to do with multiple genes contributing to both the development of complex disease traits that are associated with longevity, as well as the ability to survive those complex traits.
How drastic were the changes from 2003 to 2015?
There were very drastic changes from 2003 to 2015. 2003, remind everybody, was right around the time that the draft human genome was declared complete. Since that time, of course, there have been numerous technological advancements that have reduced the cost and increased the throughput of human DNA sequencing, facilitating the development of genome sequences now for tens of thousands of people and exome sequences for hundreds of thousands of people, which was an effort that for one individual for the Human Genome Project required approximately 10 years and hundreds of millions of dollars.
Looks like we have time for one last question. Is there any way to repair genetic mutations?
Interesting question. As you may be aware, there are numerous technologies now that have been developed to perform what may be referred to as genome editing, whereby individual mutations can either be introduced or corrected using a variety of different techniques, including increasingly now CRISPR and Cas9 technologies. Those have been considered actually for therapeutic applications more recently, and there are a variety of different companies that have espoused that technology as a therapeutic platform, including Epidemia, which is a company that we work with closely to bring genome editing-based technologies to the fore.
I would like to once again thank Dr. Dewey for his presentation. Do you have any final comments for us today?
Well, I'd like to thank everybody for tuning in, and thanks again to the organizers for the invitation to talk to you a little bit about our work. And please feel free to reach out to us if you have any questions or are interested in collaborating with us on human genetic studies.
Thanks again. This is an educational webinar and thus offers free continuing education credits. After the webinar is over and to get your credits, click on the CE button located in the bottom left-hand corner of your web page. Today's webcast will be available for on-demand viewing through November 11th, 2016. You'll receive an email from LabRoots alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. See you next time. Goodbye.